Hello, so today's tutorial is going to be on reverse phasing. In Borland Genetics, there are two ways to accomplish reverse phasing. One is using the free workflow or tool, and I have to be honest, it's quite difficult, which is why I created the subscription automated workflow. So you have two options. This tutorial is going to be on using the automated version because it's just so much easier. And then maybe once you've gone through that once or twice, you may want to try the manual version as well and learn how to do that. But I suggest starting out with the automated version because the making of the chromosome maps is pretty complicated unless you're an expert at DNA Painter, which is an awesome tool, and I recommend that you become one. But anyway, we're going to start out just so you can actually be successful at a reverse phasing project in a relatively short amount of time with the automated version. Okay, so here we are, and I'm at my home screen, and we're going to do a reverse phase project. And let's see how we do that. We go into our tools, and create a private project. We don't click on reverse phase. That is the non-automated version of the tool. I mean, you could do that, but it's kind of an expert tool if you're not using the creeper. So we're going to use the creeper and create a private project. This is a subscription tool. And we'll call the project name reverse phase sesame rice. Okay, and we're going to add a project description, phasing my mother's DNA into my two maternal grandparents using myself and my sister, brother. Okay, I don't even have a sister. I'm not sure where that came from. I guess it's getting late here. Project type, we're going to choose reverse phasing. And we don't really need to upload an image. If you want to, you can. So we're going to create a project. You've successfully created a new project entitled Reverse Phase Chesney Rice. Back to the home screen. And you'll see this is the first project in my test account here, in my demo account. We're going to go to the project now, and we're going to follow the directions. These smart projects take a little bit of time to load, especially if you have a lot of DNA kits or if you're further into the workflow. Okay, so we scroll down to the creeper. You see we've got a blank lab notebook there, and we've got a blank phase map locker. No inventory, no DNA inventory yet. So let's go to the creeper and see what they say to do.
Hello. Welcome to your new creeper-assisted reverse phase project. I'm the creeper, and I will help you reconstruct DNA profiles of the parents of our subject donor, by phasing him or her against the DNA of his or her child. There are a couple of preliminary things you should know. First, there is no need to add DNA resources to this project. To the extent necessary, I will do that for you as we proceed. Second, as we progress, it might take me longer and longer to figure out what to do next. I'm learning, so please be patient. I do my thinking every time you open the project, so sometimes your browser might spin a bit before you get to the project information screen. This is normal for now. Finally, I'll remember where we left off each time you come back. I'll also leave automated entries in the lab notebook below, to keep a record of our progress. You can do the same, incorporating your notes, if you like. If you should at some point decide to delete this project, our lab notebook will also be deleted, but all of our work will remain. The first thing we need to do is select the subject donor for our project. Eligible subjects will appear in the drop-down menu in the action pane to the right. Please select our subject donor, and then click the Activate button to proceed.
Okay, so I'm going to look here, and my mother is the only eligible donor. And that's because most of my kits are not in the demo account, they're in my personal or research account. Okay, so let's activate the creeper. Creeper successfully activated.
Perfect. We've activated a new project where we can focus our work on reverse phasing this parent-child pair. From now on, whenever you navigate to this project, I'll be there and I'll exclusively focus on our reverse phasing task.
Okay, and I guess we have 2 buttons to return to the project. I might clean that up at some point, but I assure you they both take you to the same place. Okay, so here we are back at the project, and there's the creeper. And I had already linked the father and mother profiles to my mother. They're empty profiles. They don't have any kits attached to them. But I did a run through of this, so I wouldn't have to create the profiles for you. Otherwise, you'd just be prompted to create the donor profiles, just like you do when you upload kits to the site. But as far as me and my brother, it already recognizes that those are her children. Okay, so let's see what the creeper has to say. I think most of the talking in this video is not going to be me. It's going to be the creeper.
Hello. I'm the creeper. It's time to do some phasing. Please advise me if I have your permission to create in-phase and evil twin kits by phasing the indicated child against our subject donor.
And of course, yes. So now we've got to wait for it to create the kits. But that should only take a minute or two. So just to recap here. Oh, no time for a recap. All right, let's go back and see what the creeper says, because this is how easy the reverse phase process is now. I know I made a video about how to reverse phase like a year and a half ago, and you had to use all these tools and make Excel spreadsheets and whatnot, but right now it's a bunch of clicking yeses, and then by the end it's going to have a nice result for you. We've come a long way in the last year and a half. Here we go.
I've submitted the selected donor kits for processing using the Ultimate Phaser. We should have the results back in no time. Let's go back to our project for now.
Return to project. Okay. Now what's the creeper say to do?
Hello. I'm the creeper. It's time to do some phasing. Please advise me if I have your permission to create in-phase and evil twin kits by phasing the indicated child against our subject donor.
So that should sound very familiar. It's like what we just did, except now this time the child is my brother. So yeah, let's do it again. Let's just say yes. Okay, so that didn't take long at all.
I've submitted the selected donor kits for processing using the Ultimate Phaser. We should have the results back in no time. Let's go back to our project for now.
Okay, so we return to the project. Okay, so let's see what we've got in the project, by the way. Notice that when we started, there were no entries in the lab notebook. But if you could see, the author of some of the entries in the lab notebook is the creeper. The Borland Genetics creeper has left 2 entries here. So it describes some of the things it did in the workflow, so you can remember later what you did and what you didn't do. And of course, if you wanted to add your own notes to supplement this and add more details, you can compose a new text. Or if you really don't like what the creeper said, you could delete it. Okay. If this was a collaborative project, it also lets you message the other donor. However, at this point anyway, the creeper is not returning messages. The creeper is not a real person. But if you were collaborating on a different type of project with a different user, you could also send that user a message in addition to leaving a comment here. So what do we got now? So now we've got some stuff in our project inventory. We have 1, 2, 3, 4, 5 kits in the project. So it added my mother's ancestry kit, and it added the in-phase and evil twin kits for both for her with respect to with both of her children. Okay, so now let's go back up to the creeper and see what's next.
I see you have one or more new kits processing or queued for processing. Let's wait until they are done processing before we proceed.
Okay, so processing the kits takes a couple hours, so I am going to pause the video and I'm going to come back when they are done processing, which will probably be in the morning because it's kind of late here where I am. Okay, I'm back, and let's see, I think my kits have finished processing. We can go to the resource manager, and remember there were 5 kits. Status, results ready. Results ready. Alright, so they look like they're all results ready, so we should be ready to go back to our project and continue. Here we go, reverse phase. And now we're a little further on, so it'll take a little longer to calculate what to do next. But it shouldn't take too long. Certainly not as long as the hours and hours and hours it used to take to do this manually in Excel. There we go. Alright, see what the creeper tells us to do.
Now, let's get started on chromosome mapping. Our starting point will be the Relationship Manager tool, which is directly below the Creeper interface. In this project, the Relationship Manager consists of a series of project role cards, and below that, a drop-down menu where you can select from the donor profiles of our project member kits. What I need you to do is select this project's lead donor from the drop-down. Upon doing so, a set of match cards should be displayed corresponding to the donor's strongest matches. Then, I need you to make a decision for each of the match cards as to the appropriate parental phase of the indicated matching relative. For example, if the match is only related to the donor via the donor's father, simply select paternal relative and then click the button below the match card to update the database with your selection. You will know the database has been updated because the match card will change color. 2 of the options beneath each match card require a brief explanation. First, the related on both sides option encompasses much more than double cousins. You will select this option, for example, when the match is a descendant of the donor, as the match necessarily is related to both of the donor's parents. Likewise, you will select this option for full siblings of the donor, as well as descendants of the donor's full siblings. Second, the exclude from analysis option should be used in the following situations. 1. When you believe the donor is not in fact a close relative, but appears on your list due to a defect in a kit attached to that donor. 2. When, perhaps due to endogamy, a particular match is causing conflict segments on your phase maps. And 3. When you suspect that the match might be related on both sides, and although you may not be certain, you will err on the side of exclusion, to ensure that your phase map segment boundaries are properly delineated. As a side note, our lead donor will likely have new matches from time to time, so make sure you periodically review the Relationship Manager tool in this and any other SMART project, to be sure your phase assignments remain up to date. Okay, enough of my long-winded instructions. When you have assigned phase to each match that you recognize, click the button on the right to continue.
So I've already assigned phase to all these matches, and most of them are on my grandmother's side, and rather than my grandfather's side, because I have a lot larger family. They were here in the United States a generation before the other. So You see I put in yellow for related on both sides, me and Stephen, because we're related to both of those grandparents. I mean, they're our grandparents. This one, it says private on some of these, but they're in my other account. They're in my personal account. They're just living people. But I had already gone through and did what the creeper said and assigned phase to them. And if I want to change it, let's say I found out this guy is, a paternal relative related to my mother, in relation to my mother, I could just switch it here, and it'll update the database, and it'll remember next time too. Okay, so we've done that. Let's click next. Refresh and continue. Again, it needs a few seconds to think here. Okay.
Time for some fun with colors. I found a kit link to our project that requires chromosome mapping. Let's use the Borland Genetics Here Mapper tool to create a phase map that we will later use as a mask for segment extraction.
Okay, so it wants to map one of the 4 kits it created for my mother. So let's map it. It's going to do it itself. Okay, so gives us some information here, but this is really just purely informational. We're just going to do what the creeper tells us to do.
Here we are at the Borland Genetics Here Mapper tool, version 2.0. Below, you will see a list of matches to the indicated DNA resource, sorted by position on the genome. For now, you don't need to do anything with this. Just bear in mind that the Borland Genetics database is young, so most of the matches you see will likely be to the kits of close relatives whose kits you uploaded to or created on the site. For this reason, to facilitate more thorough chromosome mapping, Borland Genetics provides opportunity to import your GED matches. In the future, Borland Genetics will also allow import of segment lists from certain testing companies. If you wish to import GED matches, you must run their Tier 1 segment search tool on this exact mono kit, or on a mono kit created on GED Match using the same process by which you created the instant kit. For many of the mono kits produced here at Borland Genetics, there is no equivalent process at GED Match. However, you may be able to upload your mono kit to third-party sites if you select compatible settings on the Borland Genetics Advanced Download form. When generating segment lists from GED Match, in their segment search settings, be sure to change the max kits to 10,000 kits, the minimum SNP count to 500 SNPs, and the minimum segment size to 5 centimorgans. Specific instructions for pasting the segment search results are provided below. Whether or not you decide to import third-party data, the navigation buttons to continue are located at the bottom of this page. If you need time to generate GED Match segment data, just click the Back to Project button and this task will be waiting for you when you come back. Otherwise, click Proceed.
Okay, so GEDmatch right now does not allow uploads from Borland Genetics, so we're just going to, otherwise if they did, you could just paste the Tier 1 segment search tool in there. I'm not sure that other sites are or aren't allowing uploads yet. That's something to talk about in the future. But we're just going to scroll all the way down to get to the Proceed button. And since I uploaded a lot of kits of family members, we shouldn't have any problem here. Okay, so now, let's let it tell me what it wants to do.
Below, I have gathered some information about your top matches in the Borland Genetics database, and I've also included your top third-party matches if you chose to import them on the previous page. As far as the Borland Genetics matches, the phase designations should be pre-populated according to the decisions you made using the Relationship Manager tool. However, now that you can see information about the individual kits to be used in the analysis, it's probably a good idea to review the designations for accuracy. Also, if it's been a while since you visited the Relationship Manager, you might have some new matches that need designation. Our database is growing quickly. For third-party matches, you'll have to make the designations from scratch. Make sure you review the written instructions below which explain the different designations in more detail. Once you are happy with your designations, click the Proceed button at the bottom of the page to continue.
Okay, so we just did this a few seconds ago in the Relationship Manager, so these are going to be accurate, but this tool also contemplates the possibility that maybe you made those selections and then like a month later you're running this again and you might have some new matches. But in the few seconds since we've just made these designations on the previous screen with those colored boxes or rectangles, nothing here has changed. But there's some unknown here. That's interesting. So maybe some of those had not taken in the database yet. So I'm just going to select them really quick, the unknowns. Eva Douse, that's Lithuanian side, my mother's side. So here we go. Proceed to the HR mapper results. Okay.
We're almost there. I've created a phase map based on all available segment data, and I've stored the phase map in your phase map locker, which can be viewed at any time from the Tools tab. I've also submitted the phase map to our project. However, I'm just a machine, and you're a lot better at assigning phase than I am. So I've also provided a button below where you can download the phase map and examine it for accuracy and completeness. Eventually, we'll have some more tools for fine-tuning phase assignment here, but I strongly recommend downloading the file and then uploading it to DNA Painter for examination. You'll want to do things like examine undecided segments that might only reflect small single-segment matches to distant relatives. You'll also want to look for small segments with no matches to see if they warrant assigning to them the phase of their neighboring segments. This is appropriate when a small gap in matches occurs between two same phase segments and you believe that the gap is due to recombinations that occurred in generations farther back. I would not recommend assigning phase to tips of chromosomes that do not have matches, unless phase can be inferred from the results of other donors. Chromosomes are generally more likely to recombine near the tips, so even a small matchless tip could have been a recent recombination. One more thing. In dealing with small single-segment matches under 20 centimorgans, I caution you to look for trends among clusters of matches, and not to rely solely on the accuracy of a single small segment. Small single-segment matches may appear to be related to you in one way based on a shared ancestor in your trees, but the common ancestor responsible for the shared segment may be farther back in an entirely different branch. Another thing. I may have labeled one or more segments as conflict segments. This usually happens due to a poor overlap situation where a false positive match makes two neighboring segments appear to be a single segment. Sometimes, you can review the matches on the segment and identify a problem kid for exclusion, and then run the here mapper again. We'll have some instructional videos on how to quality check a phase map later this year on Kevin Borland's YouTube channel. If you make any changes using DNA Painter, just export the modified map and upload it to Borland Genetics from the kit settings screen for this kit, and I'll automatically apply the most recent phase map when we do our segment extraction. When you're ready to move on, the navigation buttons are at the bottom of this page.
Okay, so if we wanted to, we could, download the phase map, and that's just going to be there, and we could upload it to DNA Painter, and we could, examine specific segments and change designations. But first, let's just take a look. Let's go to our phase map locker, which now contains this brand new phase map here. And just take a look at it. Let's get a graphical preview. So we had a lot more maternal relatives test, so it's not too surprising that there's a lot of pink as opposed to blue. Black are segments where there are no matches currently. and green are segments where there are matches, but they weren't close enough where we didn't determine side on them yet. So this is where there may be one or more distant matches. And that would be the situation where I'd want to go into DNA Painter, or in the future use some of the tools on this site to resolve these matches before proceeding to the next step. But for now, let's just go back to the project, which is here. There's a little link to it in the preview. And don't worry, I know that was really long, the description from the creeper there on what to do next, and we could just go to the next screen from now on, because we're going to do this 3 more times for the other 3 chromosome maps, and we're not going to have to listen to that again and again, because it will, thanks to Graham Hart, it's going to stop speaking when we go to the next screen. Okay, so here we go.
Time for some fun with colors. I found a kit link to our project that requires chromosome mapping. Let's use the Borland Genetics Here Mapper tool to create a phase map that we will later use as a mask for segment extraction.
So this is the second out of the 4 phase kits that it created. And remember, the Borland Genetics method is phase, then map, then extract, then merge. So first we did all the phasing, and then it had to process the kits, and now we're doing the mapping. Let's map it. Okay, and we've already listened to this, so we don't really need to listen to that again. It's the same deal. Let's proceed to the close relatives review. And again, here we go. Maternal. I'll have to investigate as to why some of these aren't taking. Hopefully by the time you try this tool, this problem will be fixed. Let's say... Getting a lot of incomings there. Okay, proceed to results. And let's just go to the locker and make sure it's there. And there's the second one. Before we only had, this is the map I did last time, or on a different tutorial for one of my paternal grandmothers. Okay, so let's take a look at the new one. Graphical preview. Okay, looking pretty good, but again, you know, a lot of segments with no matches, and that's to be expected with relatives when the ancestors were recent immigrants from Poland and Lithuania. Okay, let's go back to the project. And now that I'm not listening to the creeper, you could, if you wanted to, if you forget what to do next, it's going to go pretty quick. The longest step in each thing, I think, is just waiting for the creeper to decide what to do next.
Time for some fun with colors. I found a kit link to our project.
Yep, there we go. Let's do the third one. Proceed to the close relatives review. Okay. Let's take a look at that. Just make sure it looks okay. Graphical preview. Okay. And now let's do the fourth map. And again, there are some green segments in here that if I was taking my time and doing this, you know, as opposed to just trying to make a quick tutorial, I would go into DNA Painter and I would fix those, and then I would re-upload the map to the site if I wanted everything to be perfect, which I will do. Okay, so next.
Time for some fun with colors. I found a kit link to our project that requires
Okay, last one. Fourth map. Proceed to close relatives review. Same deal. Internal. Okay. All right. So let's go back to the project. We could have looked at this one too, but I'm just in the interest of time here moving on. Alright, so now we're back in our project and we've mapped all 4 of the phase kits. So let's see what the creeper says to do next.
I have created 1 or more special reverse phase chromosome maps and attached them to the Evil Twin kit in each in-phase kit Evil Twin kit pair. I recommend that you open these maps in DNA Painter and make sure I didn't miss anything. This map is a bit different than the kind of maps we've been making up to now. The top copy of each chromosome on the map doesn't represent the paternal copy for any donor, but rather the segments passed from the parent to the child. Likewise, the bottom copy of each chromosome on the map represents what the evil twin pseudo-sibling inherited. Once you've had a look and uploaded any corrections you deem appropriate, refresh this page and we'll be ready to continue.
Alright, so let's just take a look here. And you see it's created 2 maps called Special Reverse Phase Creeper Output. And one corresponds to me and one corresponds to my brother, because what it's in a sense doing is 2 separate reverse phases at this time. So if we want to take a look at one of these just to see what it looks like, these are a different type of phase map as it mentioned, but a picture says a lot more than the words do. See, because now it shows on the top copy, say for chromosome 1, it shows what, let's see, is this me? Yes, this is Kevin. It shows what I inherited. I inherited on my maternal copy of my chromosome stuff from my grandmother here, and then from a little bit from my grandfather, and then back to my grandmother. Likewise, my evil twin would have inherited, just exactly the opposite. And there's a reason it plots them like this, and it's because it's really easy to see the recombination points where they occurred. Okay, but let's go back to the project and see what the creeper says to do next. Again, we have to wait a little bit. And note those green segments are still there because we didn't resolve them, but if we wanted to, we could have done that. Okay, so next step.
Since we are dealing with a multiple child workflow, I will take advantage of the children's matching region data and apply logic rules that will expand some of the segments in your special reverse phase maps. Let's use the Borland Genetics Segment Expander tool to create enhanced phase maps corresponding to each parent-child pair.
So you don't have to worry too much about what that means, but what's interesting is that since there's more than one child being used, because we're not going to get, we're limited to the amount of data that my mother inherited from her parents. So whether you do it with 1 child or 2 children, it's theoretically the same amount of data that you're going to end up with, assuming you can properly assign phase to all the segments on these maps. However, it's helpful to have other siblings because in the automation process, you could use logic rules and they'll be automatically applied to help expand some of the segments and increase your coverage. So let's see what it does. And so for example, look here at chromosome 2. This was before we applied the segment expander. And you see there were 2 large black segments that had no matches, and therefore we weren't able to assign phase. However, now we look at the segment expander results, and we see that one of those has been completely resolved. Let's see, any other good examples? Yeah, look at number 14. So chromosome 14 had 2 empty segments here, and now they've been properly assigned in the after diagram, and you can see why, because on chromosome 14, when you compare me and my brother, our maternally phased kits, there were no recombinations. So therefore, if it's blue anywhere, it's going to be blue on the whole chromosome. But these are the types of logic rules that are being applied. You really don't need to concern yourself with them because it's doing it automatically anyway. But that's what's going on here. It's not any kind of magic or imputation or anything like that. It's all inferences. The type of inferences we'd make if we were like using DNA Painter like as experts. Okay, so same thing, except this is a second iteration where it's it's adjusting Stephen's chart using mine, as opposed to the first one was adjusting my chart using Stephen's. And you're getting similar types of things. This segment here that was matchless is now properly assigned to pink there. Okay, so you get the idea. Oh, look at the X chromosome too, big difference there. Go back to the creeper.
I've enhanced the phase maps associated with this project using the Borland Genetics Segment Expander tool. I've likewise stored a copy of these new maps, including intermediate iterations, in your phase map locker. Now, let's go back to our project and complete the final stage of this workflow, segment extraction and merger.
That's right. First we phase, then we map, then we extract, then we merge. Let's go back to the project. Again, it's taking a little bit longer, but still not too bad. Okay, so let's see what it tells us to do next.
At last, we arrive at the final stage of this workflow. If you are happy with your phase maps, click the Continue button, and we'll move on to segment extraction and merger.
Now, like I said, in real life, if I weren't just making a quick tutorial, I would go fix those green segments and take a look at the matches on them. But for now, I'm going to say, yes, I'm happy with the maps, just so we could get some output here and show you how it works. So let's finish this workflow. Okay.
I've gathered all the necessary resources. When you click the Continue button, you'll be shown a wait screen while I perform the necessary segment extraction. Sometimes, the wait can be as long as 15 minutes, depending on how many children of the donor were included in the workflow. However, once you click Continue, please do not close this tab or your browser until you are informed that our output kits were successfully created.
Now, right now, there's a 15-minute timeout, and that's more than enough time for a two-sibling workflow. I don't have a third sibling, so I haven't really tested it as far as whether it's going to time out or not for third or fourth or five, depending on how many siblings you have. If this turns out to be not long enough for a lot of people, then I could always either up the time frame temporarily, maybe make an appointment, tell me when you're going to do this. That way I can extend the time frame for you, because I don't really want to have users always being able to run 2-hour scripts on the site and lock up my server or anything like that. But if it's for a legitimate purpose or something, maybe we just coordinate a time and I'll extend it for you, or extend it specifically to your account or something like that. Okay, so yes, I am ready. And in the future, it's not going to be an issue, because right now these are all these are a lot of these tools, such as the ones that are being used here, are PHP tools, and I'm going to make them cloud tools in the future, which will be run instantly. Okay, so yes, I am ready. So wait for 15 minutes. Okay, it's done. So let's click on the creeper and see what it says next.
Well, we did it. I've extracted the relevant DNA data from all the segments we allocated to each of the parents of Kathleen. Since this appears to have been a multi-child workflow, there were multiple output files generated per grandparent. The final merged output kits are the ones that indicate in the notes that they were created using the Humpty Dumpty Mono Merge utility. You can find these new kits in a few different places. First, they will be accessible in the DNA inventory of your Resource Manager widget. Second, they will be accessible in the DNA inventory of our existing Reverse Phase project. Finally, my sensors indicated that you had not yet created a regular Creeper project, so I created one for you and sent the output kits to that project too. You can find your new Creeper project on your home screen, and I strongly encourage you to check it out and follow that project's workflow. That's where we'll start drilling back further by mapping the chromosomes of these new kits. Please click the Continue button to return to our project.
Okay, so in terms of the Borland Genetics method, you see it created some intermediate kits, and that was when it did the segment extraction. So that was step 3, extract, and then it used the Humpty Dumpty tool and merged the results that correspond to each grandparent into single files. So that's what's going on there. Okay, and some of the temporary files that it creates could have unusual matching properties due to a concept, I guess it's statistical entanglement or the lack of statistical independence. So eventually I'm going to have something where it will sort of self-destruct the intermediate files after a while, but I want to leave them up in your account for a little while in case you need to undo something. That's going to be a future feature. Okay, but for now it just saves them and you can delete them if you like, but don't delete the final ones, the Humpty Dumpty ones, the output of the merger. Okay, so let's proceed. Return to project. And now this is a lot of thinking again, but just a few seconds, I hope. Okay, so here we are, and that's a lot of phase maps we've done now. And look what we got here now. We've got a kit that says it's created using the Humpty Dumpty for my grandfather, and the coverage on him is 38%. Likewise, 38% of my grandmother. Okay, now if we were doing this on my father's side, and if my father was living with a reverse phase, those percentages might be different because my mother got an X chromosome from each of those, but if it were my father, he would have only gotten one X chromosome, so the percentages might be skewed. So if you see skewed percentages after you run this, don't worry, it's probably supposed to be that way. Okay, so now, well, let's hear what the Creeper has to say about all this.
Hello. I'm the Creeper. Congratulations on completing this reverse phase workflow. We have no tasks remaining here. If you need help deciding what to do next, you can either go to your main Creeper project, or you can hop to any other incomplete smart project you may have created.
Okay, so let's just do that for a minute, because now it said it created a new generic Creeper project for us. And typically what I would do if this wasn't a demo account, if this was my own account, the first thing I would do when I subscribe is create a generic Creeper project before I create any reverse phasing projects or anything, because that generic Creeper project will actually spawn the reverse phase projects and whatnot that you need for you. So you don't even have to think about which you want to reverse phase. Okay, who you want to apply the reverse phase to. But let's go to my home screen here and let's just take a look at that new project. And there it is, my Creeper project. That was not there before, if you remember. So let's see what's in it. Just them, because it's a new project. All right, so we will check back after they process, because I just want to show you the matches and show you that they behave similarly to, or just like any other DNA kit. Okay, so let's see if our results are ready. And okay, so we've got, this is my maternal grandfather, and we've got here, that was using the reverse phase utility. That was an extraction kit. This was another intermediate extraction kit. And this is the final merge kit created using the Humpty Dumpty merge utility. So let's check out his matches. And I'm not going to scroll down too far because of private names or whatnot. But I'm imagining that the first row of matches should be determinative as to whether this worked or not. Okay, so matches my mother, matches himself, which is matches the intermediate kits, matches my brother, significantly more than he matches me. I think this is my mother's sister, and this matches a reconstruction for Felix Cessny, who was my grandfather's father. And this is an independent reconstruction that does not rely on the reconstruction we just did now. Okay, so looking good. I don't see any matches on the on my mother's maternal side, which is a good thing. I mean, these are all on both sides, but I got to tell you, I cheated and scrolled down and looked at the private matches, and it looks correct. So now let's go to, and we'll be able to see that more when we look at my grandmother. because I've tested more of her relatives. Let's look at her matches. Okay, now this time I have inherited a significantly higher amount of DNA from my grandmother than my brother has. And this is my mom first, and then we got my aunt. This is my grandmother's father. And now I'll let you scroll down here a little bit because we got Lithuanian. This is my grandmother's mother, and this is an independent reconstruction for her. We've got some other relatives that she's matching in the database that I've reconstructed using cousins, using totally different methods, and these are all correctly on the Rice side, on my mother's side. And my cousins Laura and Paula are both on that Lithuanian side of my family as opposed to the Polish and Lemko side. So it worked. Okay, that's it for this tutorial. I will see you next week with a new one. All right.